Welcome to Pharmacology Insights, where we break down complex pharmacology into simple, easy-to-understand concepts. Imagine a disease that slowly steals a child's mind. Their words, their memories, and their very movement, while medicine watches helplessly from the fence. For decades, this was the stagnant reality of Hunter syndrome. A perimeter we couldn't breach. Until now. Hunter syndrome, also called mucopolysaccharidosis type 2, or MPS II, is a rare X-linked recessive lysosomal storage disorder. To simplify, it primarily affects boys because it's carried on the X chromosome. In the US, fewer than 500 people live with this condition. At its core, this is a lysosomal storage disorder. Think of the lysosome as the cell's recycling center. The place where waste is normally broken down and cleared away. An enzyme called iduronate 2-sulfatase, or I2S, acts as the industrial shredder within that center, grinding down complex sugars known as glycosaminoglycans. But in Hunter syndrome, a genetic mutation leaves that shredder broken. Without I2S, these sugars, specifically heparin and dermatan sulfate, don't get recycled. Instead, they pile up, eventually clogging the lysosomes until the entire cell becomes toxic. This cellular trash is what causes progressive damage to everything from the heart to the brain. The result? Coarse facial features, behavioral problems, speech and cognitive difficulties, heart problems, seizures, muscle stiffness, and mobility limitations. Without intervention, the disease is often fatal by the second decade of life. For nearly 20 years, the only approved treatment was idursulfase, marketed as Elaprase. It's a classic enzyme replacement therapy. You give the missing enzyme back to the body through a weekly intravenous infusion. It works, for the body. But it couldn't save the mind. The brain is guarded by the blood-brain barrier. It's a tightly regulated wall of specialized cells lining the brain's blood vessels, designed to keep toxins and pathogens out of the brain. Unfortunately, it also keeps most medicines out. Standard enzyme therapies, like idursulfase, are too bulky to pass. So while the body improved, idursulfase could not address the neurological symptoms of the disease. Cognition, behavior, and development continued to decline. This is where Tividenofusp alpha, brand name Avlayah, enters the story. This isn't just a new drug. It is a new delivery system. So, how does Tividenofusp work? The brain has a front door for iron using a protein called transferrin. The blood-brain barrier has receptors specifically designed to grab transferrin and pull it across, in a process called receptor-mediated transcytosis. Essentially, the cell grabs the molecule at one side, engulfs it, carries it through, and releases it on the other side. Developers of the drug essentially built a biological Trojan horse. To do this, they took the I2S enzyme and fused it directly to the top, at the N-terminus, of an engineered antibody fragment. Specifically, a stripped-down Fc region with no Fab arms at all. This is not a conventional antibody. The Fab arms, the parts that normally recognize and grab antigens, were removed entirely, leaving a lean, two-domain Fc scaffold. The lower portion of this scaffold, the CH2 domain, has been deliberately silenced so it cannot trigger immune effector functions, reducing the risk of inflammatory side effects. The real engineering marvel sits in the CH3 domain, where scientists used directed evolution to re-engineer a small patch of just 9 amino acids into a loop that binds the transferrin receptor, the brain's own iron import gateway. So the final molecule looks like this from top to bottom. The I2S enzyme riding on top, a quiet CH2 domain in the middle, and a CH3 domain with a custom-built grip on the transferrin receptor on its sides. The trick is that the brain thinks it's importing iron. The reality is that it's pulling in the life-saving enzyme across the blood-brain barrier. via receptor-mediated transcytosis. Once inside, the enzyme is released into neurons and glial cells, enters their lysosomes, and starts doing its job, breaking down the accumulated heparan sulfate and dermatan sulfate. In plain English, the scientists essentially disguised the I2S enzyme as something the brain already knows how to import. They attached the enzyme to a molecule that hijacks the brain's own iron transport system, using the brain's front door, rather than trying to knock down the wall. For the first time, medicine is cleaning up the waste inside the brain, not just in the rest of the body. So, does it really work? The FDA's accelerated approval points to yes. In early to mid-stage trials, involving 47 pediatric patients with Hunter syndrome, the results were striking. At 24 weeks, heparan sulfate levels in the cerebrospinal fluid plummeted by 91%. To put it simply, within 6 months, most of that toxic trash had been cleared. Even more promising is the impact on the neurofilament light, or NFL chain, a protein that leaks into the system when nerve cells die. Basically, a marker of neuroaxonal injury. By week 104, patients saw a 70% reduction in NFL. This suggests that the drug isn't just clearing waste. It may be slowing down the nerve damage that defines the disease's neurological progression. Early gains in adaptive behavior and cognition have been observed, though the ongoing COMPASS trial will provide the final, confirmatory word. Tividenofusp is approved as a once-weekly intravenous infusion. It is intended for pediatric patients before advanced neurologic manifestations set in. The timing matters enormously. This is not a rescue drug for end-stage disease. It's designed to intervene before irreversible neurological damage sets in. On safety, the labeling carries a boxed warning for allergic reactions, including anaphylaxis, so infusions must happen in a monitored medical setting. Clinicals should also watch out for anemia, because the drug interacts with iron transport. Kidney function should also be monitored for potential nephropathy. The approval of Tividenofusp is described as a milestone in Hunter's syndrome. This is the first FDA-approved biologic to successfully hijack the brain's transport system. But the significance here is bigger than one drug, or even one disease. If we can sneak an enzyme past the blood-brain barrier for Hunter syndrome, why not for Alzheimer's? Why not for Parkinson's? Tividenofusp didn't just cross a biological wall, it broke a glass ceiling in pharmacology. It proved that the most unreachable parts of human suffering are finally within our grasp. That is the definition of a breakthrough. And that's it for today's pharmacology video. If you enjoyed this video, please give it a thumbs up and subscribe to the channel for more pharmacology content.